Well, thank you all for coming, and it's an honor to be here to represent Southern Miss. My name's Hao Shu, and in the next few minutes, I hope to explain to you what is TNF, what are mast cells, why it's important to study the regulation of mast cells, and update you on some of the important findings we have recently. Okay, so TNF stands for tumor necrosis factor. It was initially identified as a secondary molecule that could kill cancer cells. So it was very popular for a while, but later people found out that many cells, almost all cells, will produce TNF receptors. So by binding to those receptors, TNF could mediate a number of pathological reactions or physiological responses. My lab is particularly interested in mast cells, which is best known to cause allergy. Okay, so turned out that mast cell also is a major producer of TNF, and mast cell-derived TNF is responsible for mediating immunity against bacterial and viral infection. It's also is important to recruit and activate T cells, which is by itself important in the organization of secondary lymphoid organs. But when overexpressed, mast cell TNF is known to cause neuronal inflammation and neurological pain. And what's not on slide is that TNF derived from mast cells could lead to allergic airway diseases. Okay, so the current therapeutic approach to TNF-linked disease is using anti-TNF biologics, okay, basically inhibit the overall activity of TNF. And it works really well for rheumatoid arthritis, but it doesn't work that well for the other TNF-related diseases, including mast cell TNF-related diseases. And also we can tell it will cause a lot of side effects. A lot of patients who are under that treatment will have a high risk of infection. Okay, so we think we can explore a better way by studying the regulation of mast cell release of TNF. So unlike other TNF producing cells, mast cells could pre-store the TNF in those secretory granules. So when mast cells, for example, are under allergic conditions, those granules could undergo this process called degranulation to release the TNF. And also active mast cells could produce TNF, and newly synthesized TNF could be released through the secretory, the classical secretory pathway, basically from ER to Golgi to the secretory vesicles. And then when the TNF is translocated onto the plasma membrane, this enzyme could activate it and cleave it, so the soluble TNF can be released. So we've learned that, and with others, that the allergic conditions, the mast cell could release TNF through both pathways. But when TNF are activated by pathogens, they only release through this pathway. So we have some data over here. And this is to show allergen and those pathogen-related molecules can stimulate the release of TNF as detected by ELISA. But if you treat the cells in the same way and look for beta-hex release, beta-hex is a marker for the granules, the allergens can trigger beta-hex release, but not the pathogens, okay? So conceptually, it is possible to kind of downregulate the TNF release from this pathway, so we prevent allergic reaction, but we can still preserve the host's ability to mediate response to pathogens. Does that make sense? Okay, so we believe there's a family of proteins that's used to differentially regulate these pathways, and those are so-called MUNC13 proteins. There are 5 MUNC13 proteins in mammalian cells, and in mast cells, there's 3 of them. There's going to be there's MUNC13D, BAIP3, and also MUNC13A or DB, depending on specific mast cell type. And we have some confocal over here to show that those TNF-positive granules are actually surrounded by MUNC13D, and we believe that the different forms of ONC13 could regulate the release of TNF through those different pathways. Okay, so how would you go study the release of TNF from, say, the granules? Let me go back to that slide to show one more data piece. We believe those granules are actually multivesicular bodies, okay, because TNF is co-localizing with the markers for multivesicular bodies. And it's not just CD63, but also CD9 and CD881. So what means is that those TNF could possibly be inside those granules on vesicles that are called exosomes. Okay, so how do we measure release of TNF from those vesicles? We take advantage of the topology of TNF, which is a type 2 membrane protein. So when it's synthesized and deposited onto the secret pathway, the N terminus will be facing the cytosol, okay, and the C terminus will be facing the lumen of the vesicle. And when fusion takes place, so the C terminal part will be cleaved and released, but the N terminal part will remain inside the cell. So imagine if you tag the N terminal part of TNF with a fluorescent marker, a protein, the fluorescent signal will remain inside the cell, right? Now, when the TNF undergo the unconventional secretion, so here the N terminus is still facing the cytosol, the C terminal is facing the lumen of the multivesicular body. But when those vesicles form through invagination, the N terminus will facing the lumen of those vesicles, those nanovesicles. Now, when NVV fused with the plasma membrane, now you have the full-length membrane-bound TNF released in association with those vesicles. Okay, so the signal, if the fluorescent tag is there, the signal will be detected in the supernatant. Okay, so that is the method we use to measure the release of TNF from this unconventional pathway. And this work is done by my student Tulu, who is over here. And she also looked at the secretion of this fluorescent signal under allergic conditions. So in resting cells, there's no signal detected. And under the allergic conditions, you see the release of this fluorescent signal in the supernatant. So we can also harvest the vesicles, and this is done by cryo-EM in collaboration with a team at LSU. So we can look at the size of those vesicles. We have also done mass spec to look at the composition, the chemical composition of the vesicles, and we found M-Sherry TNF, CD683, LAMP1, and other markers for exosomes. So do they have signaling capacities? You know, are they able to affect the target cell? So in this study, Tulu incubate those vesicles with raw cells, which is a type of macrophage cell line, and look at the regulation or gene expression of some pro-inflammatory mediators, IL-1 over here, IL-18 over here, and INOS is over here. So she finds something quite amazing, right? So you actually have a down-regulation of the expression of this pro-inflammatory molecule, but over here you have an upregulation. Okay, so that's where we are. We are in the process of trying to sort out if this effect is indeed mediated by TNF, or some of the impact could be mediated by other mediators that's carried by those exosomes. So with that, let me conclude by summarizing what I shared. So pathogens and allergens seem to explore different pathways to release TNF from mast cells, and membrane-bound TNF could be released in association with those nanovesicles to mediate cell-to-cell communication. And we have this original fluorescence-based secretion assays that could be adapted for high throughput screening for inhibitors of exosomal TNF. So with that, I would like to thank the founding agencies. I would like to thank my collaborators, but most importantly, my student Tulu, who is also here. Thank you very much.